My name is Harleen Saini. I'm a graduate student in Melissa Moore Lab at University of Massachusetts Medical School in Worcester. I have always been very fascinated by biology. My father is an OBGYN, and I was surrounded with births happening all the time around me. And I saw so many babies being born. I was very fascinated by how exactly does from a single cell a whole organism get made. I consider myself lucky to be here today at UMass in America. Where I grew up in Amritsar. It's a small town. It's at the border of India and Pakistan in northern India. I went to a very good high school. It was an all-girls school. The good thing about going to an all-girls school was that I could be fearless, independent, and bold and not worry about society's rules. And that helped me push the boundaries that the society sets for us and just be as ambitious as I wanted to be. A part of that ambition was to apply to Johns Hopkins in Baltimore. Coming to Johns Hopkins opened the doors to many more opportunities for me. I was in the environment where the best research was being done, and I could reach out to people that only existed in textbooks, but now suddenly I could actually meet them and talk to them and shake hands with them. I was surrounded by really great scientists, and it strengthened the passion that I had for science even more. Melissa Moore is one of those researchers from my textbooks, and here I am learning from her and earning my PhD at one of the best places in the country to study RNA biology. So the kind of work that we do in Melissa's lab is very basic, asking very basic biological questions. We've all heard of DNA, which contains the blueprint of life, as we say it, and RNA is something that works as a messenger between DNA and protein. Before any protein gets made, the DNA has to make a copy of itself, a small print of the particular gene that is ultimately going to make the protein. That little copy is the RNA. We want to know how gene expression is regulated in neurons under conditions of when the neuron is activated or when it's not activated. And we do that by studying the RNA in neurons. Studying RNA is very exciting, and to be at the very cutting edge makes it even more thrilling. Sequencing technologies, we hear about genomics and how you can sequence all your DNA. Not only can you sequence all your DNA, you can also sequence all the RNA in cells. And that just opens the doors to making us able to answer so many more questions that we couldn't even think about 10 years ago. I wake up every morning feeling very excited because a lot of work that I'm doing right now is computational. I can literally go on my computer and write some code and ask questions from my data and I get answers. I learn something new, and I can learn something new pretty much every day. And many of those things that I learn on a daily basis are things that we already knew, but when I see it with my own eyes, I believe it even more. I know that, okay, that has to be true.